So thank you. It's my distinct pleasure to introduce our next two speakers for this session and Fireside Chat at Innovation in Genome-Based Medicine. Dr. Olivia Adonis, I'm just going to point out a couple of highlights of their bios here, is the Chief Scientific Officer of REGENXBIO. Prior to REGENXBIO, he was Senior Vice President of Cell and Gene Therapy at Biogen, where he led the company efforts in dedicating and identifying and developing new technologies for gene transfer and genome editing. Dr. Danos is also co-founded and is an executive member of the board of directors of Lysogene, a company focused on the development of gene therapy products and candidates for the treatment of MPS type 3A. Jim Wilson is the Rose H. Weiss Professor and Director of Orphan Disease Center and Professor of Medicine and Pediatrics at the University of Pennsylvania. He's also the Director of the Gene Therapy Program at the Perelman School of Medicine. He's on numerous scientific advisory boards, including Solid GT, Dimension Therapeutics, and REGENXBIO. Jim began his career about 30 years ago at the University of Michigan, where then he moved to Boston. And if I have this right, that's at the point where both Olivier and Jim were in Richard Mulligan's lab. Do I have that right, as postdoctoral fellows? So they met, if I'm doing my math right, about 30 years ago. Jim went on to move to Penn to create the first and largest academic-based program in gene therapy. He's published over 550 manuscripts, reviews, commentaries, and editorials, and is an inventor on over 117 patents. He was the second president of the American Society of Gene Therapy and has received numerous awards in the sector. On a personal note, he was my postdoctoral advisor for about 10 years. So I welcome both Olivier and Jim to the stage for this fireside chat in innovation in genome-based medicine. And Olivier, I'm going to hand it over to you.
Thank you, Sarah. Can you hear me? Yeah. Welcome, everybody. So we're going to have a discussion. At some point, we're going to open for questions. We thought it would be a good idea to have an interaction with the floor. So the theme here is about innovation in genomic medicine. So maybe we should narrow a little bit genomic medicine because it's a very wide term. So I think what we're talking about today here is basically cell and gene therapy. It's about reprogramming cells, so anything you do to a cell to change its information would fall under this. Okay, so it's obvious to say that what's being done is either gene augmentation, replacement, or gene inactivation. And those are the technologies behind that, allowing that to happen, are generally the gene transfer technologies. And they are now married to other technologies. You bring in the cell not only a cDNA as we used to do in the old days, but much more sophisticated components that allow you to, for instance, do gene editing or shut down RNAs, etc. So it's a rather complex set of technologies that's based on decades of innovation now, and we're going to try to discuss what's the nature of this innovation, right? And how is it still happening? Are we in a phase that we've put the innovation behind us and now we have all these things happening and that's over? Is it? Are we also looking at a regular type of technology, or is cell and gene therapy something else because it's fundamentally different from what other therapeutic modalities that have been developed before? And that's something that Jim has been doing quite a lot of thinking and research around, so he's actually the ideal character to try to question on that. So I think we'll in the first part of the discussion, I'd like to maybe look at the landscape as it is today. What's happening? What are the important inflection points that are coming up? And I think we touched upon that just before here, but we'll come back on those, trying to also from that trying to describe what we or what Jim believe will be the trajectory immediately. So that's kind of the first set of questions that I'd like to ask to Jim, and then we'll get into the discussion of what's innovation in cell and gene therapy, and what's the dynamic of that? Is it still needed? Is it still happening? And so what's going on there? So I think we should just jump in.
Right.
So I think the first question is probably, what's going on? So if you look at the recent events, successes or not so successful events as well, how do you see the trajectory for the next couple of years? Let's start like this.
Okay. Well, thank you, Olivier. Before I answer your question, I do want to thank ARM for inviting me here. I'm sort of new to the organization and learning about what their goals are. But as I've studied this technology, both from a scientific standpoint and on the Penn campus with my colleagues at the Wharton School, which I've done over a couple decades, the goals of ARM, I think, really are addressing what is going to be a critical path to our success. I think the current technologies will yield some successes. I'll get back to that in a minute. But as I tried to conceptualize what our challenges are before us, I became somewhat of a student of a Harvard business professor called Clay Christensen. You may have read about his work. He wrote The Innovator's Dilemma, and he studies what are called disruptive technologies, and we are disruptive. What Clay has suggested, however, is that we have to do more than create the technology. We have to create the innovation, and what the critical path there are the sort of the ecosystems that need to support our efforts, including what are the business models. So the goals of ARM to try to bring the community together around that are probably more important than what we're doing in the lab right now to make commercial successes. So we're there with you. Keep pushing that agenda. It's critically important. Getting back to the science, we met 30 years ago, shared a bench when we thought that gene therapy would be something we wanted to do for our career, and it's been a long journey for sure. And how we got here and how we could have done things better or differently, I guess, is not something I want to focus on today. But the fact is that I believe, and we're seeing that there will be successes. The SMA study out of Avexis is an example. Some of the hemophilia data look very encouraging. But as I think about it from those days when we were postdocs in the 30 years, I think we're only at the beginning. And until you start generating positive clinical data, you don't really know where you are. I'm really excited about where we are, but I'm also concerned that the expectations are that every time we do an experiment in these R experiments, it's going to work, and they're not all going to work. Hopefully we can do this in a way in which patients won't be harmed, but this is truly experimental medicine. And the community needs to get around that paradigm of not only bench to bedside, which is what you all are doing, but bedside back to the bench, because there will be iterations. And it's interesting, some of the iterations that will be necessary to make this successful are not fundamental, innovative, new paradigms of delivery. They're tweaking the system. We don't really deal with issues of formulation. There's very little work around what is the optimal way to deliver a vector with respect to its concentration, its route, the device, its excipients that are part of that. And then there's the broader area of are there going to be immune challenges or immune toxicity, and how can we mitigate those? So my hope is as we move into 17, which we are, and I think the expectation is sort of a escalation of really interesting clinical data. Let's not panic if everything doesn't work. That's really where the field is. But we need a way in this whole sort of partnership of the academy, pharma, and biotech to not only get the products out into the clinics, but to learn from that and then stay focused in the lab to try to address what some of these challenges are going to be.
Yeah, can I maybe add something here, which is that the question very often is, why doesn't it work? Is it is it because things can not work in any industry, right? When you're making cars, there are failures as well, right? But are we talking about the same nature of failures, that is, engineering failures, or are there failures because we are missing essential knowledge or essential knowledge of what we're doing in the clinic?
Yeah, I've been involved in discussions about how you define failure, so that's probably not the right word, because
right,
as the
you
way
learn a lot from them.
Right, as the way a scientist views an experiment may be different. Until we have complete data sets of small, large animals and humans, it's hard to know how predictive the animal models are. And for in vivo gene therapy, there's a lot more work in animal models than in ex vivo gene therapy. And what I hope to see out of 17 are broader data sets where we do have small and large animal data as well as humans to try to determine what was predictive as we go through iterations. And unfortunately, in large animal models, there aren't many disease-based models, and a lot of these questions are really directed to issues related to the underlying disease. So, for example, in liver gene therapy, do we have vectors that are going to be adequate to treat liver-based diseases? And it comes down to how efficiently can we repopulate hepatocytes with the transgene, and how much of that sort of transduction do you actually need for success? If we had a vector right now that we could inject in and 100% of the liver cells were corrected stably, we wouldn't be having this discussion right now about the nuances of non-success. But we're somewhere south of that. And for example, there may be some diseases where the technology is adequate based on the delivery efficiency, but you'll only know that really in an authentic animal model or not. And I think that's where we are now with the technology, that it's advanced to the point where there's really a good chance that in a number of these diseases we're going to succeed. But let's just talk about delivery. We're not done. I mean, am I confident that we have a vector that you inject, you get 100% of the liver cells? No. How about an intrathecal delivery? Can we transfer genes into 95% of the neurons of the cortex? No. And so what we're doing is we're dealing with delivery technology that would probably be adequate in some applications, but in not all of those applications. And when you're on one side or the other of, quote, success or failure, that's really, you've got to play that out in either a really good animal model and then make a decision about proceeding forward, or you got to get into humans.
Okay, so clearly delivery is not perfect, and there's more to be engineered and discovered there. But beyond delivery, we also meet resistance with the immune system, for instance. Right? So that's another barrier, maybe that's kind of unsolved. Definitely not perfectly solved, and that everybody's going to meet. And in a way, the more efficient you are at delivery, the more problem you're going to get with an immune response eventually. So, so people could say, well, we're used to that. We're injecting recombinant proteins to patients, and we can deal with the immune response. That's okay. There are ways of doing that. But when you talk about gene transfer, what you're doing is very different, right? And the kind of immune response that you're going to trigger might be very different and is actually not very well known. Would you agree on that?
Yeah, I mean, look, I understand what can go wrong with an immune response throughout my career and some of the work that we've done, and clearly primates are different than smaller animals. And I do think for in vivo gene therapy, immune toxicity is something that is going to surface, but I think it's solvable. When the field back years ago, the field was working on non-viral adenovirus and this new gene vector system, AAV. We were in the middle of all of that, some vector systems were more efficient than others, such as adenovirus was very efficient, but it was very immune activating. In AAV, some of you may not know this, but it's a virus that has very unusual properties in that it is not immune activating, it is immune tolerizing, that it actually suppresses an immune response. And so right around the turn of the century, when the field was at this point, a pivotal time in what direction to go, many of us got behind delivery technologies that were not immune activating, although not terribly efficient, and that was AAV. And it really is a remarkable property. When you inject it, you don't activate inflammation, which is the first step to a T cell response or a B cell response. And there's good data really into humans that it not only puts the gene in, but it somehow suppresses an immune response. Having said that, there's a threshold for everything. And I suspect with AAV gene therapy, as you said, as the transduction efficiencies improve, or you introduce the vector into milieu where there's inflammation, due to underlying pathology of the disease, immune toxicity is going to surface. At the end of the day, it may require, I've always told my lab, if we have to give a drug to suppress the immune system with our vector, we failed as vector engineers. So we should be able, and I think I'm probably wrong about that at the end of the day, there may be regimens where there would need to be some sort of use of some immune modulatory drug, hopefully not permanently, but transiently, in order to just kind of put us below that threshold.
Okay, I think at this point we should maybe pause and see if there are questions from the floor. Anybody wants to comment on anything, or shall we just move forward? Luc?
Luc Vandenberg from Harvard and former mentee from Jim. Question about consolidation versus competition looking forward in the field. Kind of an open question. Do you feel like we're getting to a stage where competition may be hurting the overall progress in the field towards success, and that there is a need towards consolidation? Or do you feel that the trajectory where dozens of companies are sprouting up based on individual technologies is a trajectory that is actually going to support the field in the long run? And I guess maybe to bring some specificities, a genome editing company doesn't have delivery. A delivery company doesn't have genome editing, but maybe that perfect drug is a combination of the two. And you could probably say that about many aspects of what we're trying to achieve, which is delivering a complex therapeutic.
So those are two different C words, competition and consolidation. What you're referring to, the way I think about consolidation in a positive way is aggregating assets so that one can move forward with a more complex biologic, and that's probably the most important issue right now, is to marry the genome editing secret sauce with the best delivery in a group that can translate, and that be good to do that for sure. I've thought a lot about that, actually tried to stimulate that. There's some challenges. The valuations of the genome editing companies are somewhat of a challenge. Not for them, probably. Good for them. But that's been hard to do. And who will do that? Luke, I do think it's important to do. Maybe it would be a pharma that may end up doing that, bringing those together. But they desperately need to be brought together, or somehow the know-how needs to come under one roof, for sure. In terms of competition, I would encourage competition where it's true competition, because we're only just starting. And it would be a shame if there was consolidation of competitors right now. The academics carried this field. That's why we're here. Not just me, but if you look at the period from 2000 to 2012, where the investment was, and if you look at the successes, they all came out of academic organizations, and there is room for that. And out of those will sprout startups from smart scientists. I can tell you it's fascinating in my lab because I've got a pretty active, energetic lab, and Luke was there at one time. And we partner with a number of different companies in translational projects, but I got some incredibly smart MD, PhD students. And after a lab meeting, and we have discussions, and they're in lunch having coffee, and they're just going at it. They're not talking about the science. They get energized about some of the commercial business challenges. And so there's a new wave of talent in the academy that is more savvy about partnership and commercialization. And I think I have not seen that in my career, and I think the field needs it. And let them go do their thing and start their companies and compete and go at it, and let's see who the best, who's going to win.
Okay. Yes. Maybe you can wait for the mic. Just thank you.
Good morning, Barbara Nelson. I apologize if you've talked about this already, but I'm wondering if you could comment on non-viral vector approaches like Sleeping Beauty and Piggyback and the value of these alternative approaches to the field.
So the question was non-viral?
Non-viral.
Such as Sleeping Beauty and Piggyback.
Yep.
So I guess those are non-viral.
Transposase systems, yeah.
I think about the delivery and then what's in the delivery vehicle, so you can package things into.
Right. So they allow you to have larger packages and sort of focused insertion sites. Presumably, the value proposition is not disrupting something that may later turn into a different kind of cancer. So larger package size, more precise insertion, and then removal, if you'd like, as a way to put genes of interest in.
Sounds good.
Yeah, it sounds good, but it doesn't really solve the delivery problem. But that's an old discussion. I mean, you still need a delivery vehicle, whether it's viral or not, and the piggyback or the transposon technology doesn't really solve that. It has a lot of good features, but it's not a global solution.
It does address a question, though, that I've been thinking a lot more about recently for in vivo gene therapy for chronic diseases, and that is how can we affect a truly durable, stable genetic manipulation or whatever. So far, much of the energy has been focused on AAV, where the genome goes in and this magically forms a circle and then persists for the life of the cell. But if the cell divides, then it goes away. And there are really, in our lab, we've got durable expression for 12 years in dogs and monkeys. But I am really coming around to the notion that maybe the best way to achieve durable, sustainable expression is through a genetic manipulation. And we got a ways to go, but part of the problem when you put the gene in without manipulating the genome, you wait for things to settle out in terms of what the permanent graft is, and it may be less than what it was initially. And if you could follow me with this, if you could somehow through that initial engraftment then make a permanent change, it may be a better way to go. And so I'm slowly coming around to genome editing, I guess you'd say.
Yeah, so, okay, maybe, so since we're talking about genome editing, maybe we should stay on this topic, right? Mm-hmm. And also, at the same time, come to the second part, which is more about focusing with innovation. So we'll go to genome editing, but the general question is more, are we talking here about a mature field that doesn't really need much innovation? I mean, things have been done, or are we fully still in the innovation? And how do you see genome editing in this context, basically? Yeah,
there's one direction that I see happening that would require more innovation, and it's something that is sort of logical, and that's moving from the rare diseases, significant unmet need, loss of function, to gene therapy as a delivery vehicle for more common diseases. And an example, we have a partnership with Janssen where we're using AAV, it's funded by the Department of Defense, to squirt it in the nose and engineer the epithelial cells of the nose to express an antibody to prevent flu. Well, that's not a genetic disease, and I think that naturally we're going to move. I mean, if you follow the disruptive technology scholarship, you begin in these niche diseases, and then you move into more less niche diseases. It'll deal with the business model for sure if there are potentially 500,000 patients rather than 50. But we need to do some things to get there. And the one area that the field was full on 20 years ago, and they've just left it, and that's, can you turn a gene on and off? And it used to be sessions at ASGCT. We had like 2 or 3. And now there isn't barely an abstract about, could put a gene in and could you queue it up and queue it down? And I think that's an area that the field needs to get back to because it could provide a significant safety advantage. Because one of the reasons, in the United States, we cannot do phase 1 studies in normal subjects with gene therapy. I mean, maybe we can, but according to the guidance, it's discouraged. One of the reasons is in these settings, our form of gene therapy, you put it in and it's there for a very, very long period of time and you can't shut it off. So that's an area that I would encourage the field to get back to, and I really can't understand why. There's some technology out there that could be useful. My experience is when you propose it to a drug developer, it's just too complicated at this point in time, and there are too many sort of moving parts.
It's another layer of technology that you add to the vector, to what the vector brings in, and right. so yeah, it starts to be mind-boggling.
But that is a game changer. If if some company or group had a way to put the vector in, and then give a pill to turn it on, and then if you didn't want it on, you don't take the pill. I think that would change the way in which at least some of the applications could proceed forward with. People talk about delivery. I think, in order to deliver to some of the more challenging targets like the neurons or the cortex or maybe all of the muscle cells, maybe we have a vector. I think there needs to be some room for innovation, but if you look at AAV and you continue to tweak it, which we all are, we're going to reach an asymptote with that technology.
Are we there yet, do you think?
I don't think we're there yet. Not at all. It's easy to engineer 10 million, 100 million variants, but how you select out of that haystack the needle that's going to work in a way that you want it to work without the immune toxicity, there's no scaling of that because you got to vet that in monkeys. So that's
yeah. But there are signs that you kind of keep rediscovering the AAVs that were selected by evolution a little bit, right?
Most of them aren't any better than what we started with, but there will be. I think there will be, but there will be a limit. And I don't see that as a game changer, as what's going to catapult the field into the next generation.
Yeah. Well, so maybe let's say a few words about non-viral vectors, and I think they're back in the limelight mostly because of gene editing. So what's your take on that?
So yeah, I mean, when I left Mulligan's lab where we worked with retroviruses, I decided it was going to be non-viral vectors. So I started my lab and worked on complexes of DNA and protein, not lipids. These, actually now we call them nanoparticles, I guess.
Yeah, that's
right, so I started my career in non-viral, and I almost prematurely terminated my career focusing on non-viral. So that's it's challenging because you end up recreating a virus. The one thing we don't have with a viral system is a good transient system, which is what we would need often, I guess, in the applications of genome editing. So now that we now call them nanoparticles, or just protein delivery, definitely would be incredibly helpful for transiently expressing an endonuclease that causes a double-stranded break, for sure. My experience has been, through the field of in vivo gene therapy and some of our own experiences with the current technology, is it's still pretty pro-inflammatory. I'm not convinced that there's a system out there right now that could deliver the level, the efficiency and the level of expression that one would need. And so I don't see it right now. Someone may have it. In our hands, we haven't seen it, but I suspect we'll get there. But just one other point about that. In our experience with genome editing, which we're doing without, pretty much off the radar screen, our experience has been that the level of expression that one needs of the nuclease, whatever it is, to achieve editing in larger animals is higher than what you need in vitro. So, so I'm concerned about what not only level of transfer, whether it's a protein, this notion that I think you can get a whiff of Cas9 and you've got your editing and you're done, we don't see it. So, so I don't think we're there yet. And so far, protein delivery or non-viral, at least when you advance into larger animals, we're not there yet. And I suspect at some point we're going to be there, but not that I've seen. And I got to admit, if someone had it, I'm sure we've been reading about it in Nature or Science.
Yeah. Well, I mean, there are programs with that and people developing clinical products claiming that this is possible. But what you're saying is that it's probably still difficult to reproduce. I mean, it's not there's no standard. It's just something that's happening as we speak, right?
Yeah,
right.
But for genome editing, but even then, whether it's a nanoparticle or not, I mean, just look at Alnylam, who is a company that I have tremendous respect for, who got behind a delivery challenge, selected the liver because the liver is accessible, and it's liver-based, and really went through several iterations of their delivery vehicle and stayed with it. But the delivery of those, even outside the liver, because of the pores that are in the capillaries, is a challenge.
Yeah. Okay, so maybe in the last few minutes before we reopen for questions, can we come back on this disruptive technology notion and why you think that gene therapy is different from I don't know, monoclonal antibodies or recombinant proteins or antisense oligos? Or is it different, by the way?
Well, this is something that I've studied with my colleagues at the Wharton School, and we've tried to compare the trajectory of gene therapy to another emerging new technology, which is therapeutic antibodies, for example, in which they tracked over time, but then diverged, and it does seem to be more disruptive. I mean, when I first moved to Philadelphia and got to know some of my neighbors who were senior executives in the pharmaceutical industry, they told me that if you can't swallow it, you can't sell it as a therapeutic. Now, obviously, that changed, and that was disruptive, sort of, at the time. But the disruptive nature of this is not well. Okay, we've overcome the fact that you may have to inject something, but now the problem is, can you sell it if you only have to inject it once? And the one and done problem, which I know many of you have thought about probably much more than I have. But I did think the field suffered from a couple things different from monoclonal antibodies. One was we got way ahead of ourselves before 2000 in probably would be a classic of the so-called irrational exuberance about what this field could do, which therapeutic monoclonals didn't really sort of follow that trajectory, and then there was the decline in support. But I really come back to what ARM is trying to initiate discussions around business models, and I think we as scientists in your labs and your companies have to show this works, and we're getting there. But you have to find a way to sell it and to continue to encourage the investment, which is just phenomenal. This investment. I tell you, the other concern I have, running the Orphan Disease Center, is, and now this is a little more forward-thinking. Rare diseases are sort of the crucible by which you begin to test this. But what if we're successful and that we can move into more common diseases? I think the combination of one and done in really low markets, I unfortunately, and this is my own sort of personal concern, is that rare diseases may be left behind, and they are the diseases that are most likely, would benefit early from that. And maybe that wouldn't happen, and maybe movement towards more common diseases is going to be slower than I think. But I think not. I think as we gain more momentum in the clinic and more experience, that clever drug developers, physician scientists will find applications in which the markets are not just 20, 200, 2,000, 20,000, 200,000, and that's where that investment is going to be directed to.
Thank you. So we have a little time for questions, if any.
Here's mine.
Anyone wants to? Yes?
Mary Laughlin, coming back to your comments on disruptive technology and also this aspect of turning a gene on, turning a gene off, and the current technology with direct gene editing, I'd like to hear your thoughts and comments on utilizing the cell's capability to regulate gene expression, particularly long non-coding RNA and those types of technologies where you would avoid some of the safety concerns of direct gene editing.
Yeah, I mean, you mean sort of endogenous regulated gene expression? Boy, that would be pretty good. It's just that, and you may have thought about this more than I have, but we used to try to develop systems so that the gene would respond to an intracellular cue, so it would cue it up and cue it down. And it turns out that the collage of signals that need to come to bear to reconstitute authentic regulated gene expression are not just easy. They're too complicated to manipulate in any one kind of intervention. But if we can encourage rethinking of this regulated gene expression, getting back to using endogenous cues would probably be a good thing to do. Yeah, for sure.
Maybe a quick last question, or yes, down there. No, just use the mic.
From Lonza.
Hi, this is Sudha from Lonza. My question is that when we are looking into gene editing or the microRNAs, various different technology for silencing the gene, are we looking into the downstream implications? That is, epigenetics plays a major role, imprinting patterns, etc., and they do change. So I never come across where these two get together, where we are looking into the gene editing, but what are the downstream implications? How these imprinting pattern changes? Is there a reason?
You mean, so the question was when we modify a gene, let's say we ablate it or
silencing
suppress
it or deleting it.
Does it have consequences, some sort of cascade or downstream consequences to the cell?
Yeah, in terms of especially the methylation pattern, because at the end of the day, the DNA is DNA, but it also gets the secondary stamp of imprinting. How those changes?
Yeah. Well,
thank you.
I think we look at it not on a molecular level. We look at the health of the cell, and then at the end of the day, when we decide whether we're going to develop that as a drug, we conduct safety testing, and it does raise a question, though, about analyzing the implications of a genetic intervention when we have things such as single-cell RNA-seq. we could generate loads of data, and I think individuals are, which at the end of the day, though, we have to determine whether there's any functionality of that. It's sort of like looking at integration events, in the spectrum of integration events in an ex vivo gene therapy experiment. What's really important is, do they have consequences in terms of oncogenesis? So those experiments will be done, but just a cautionary note, just because it looks like there may be an upregulation of p53 twofold doesn't mean that that's a non-starter. We have to determine whether the upregulation of p53 has consequences. So we need functional assays, and that's regulatory science, and that's going to be an important part of the evolution of the field.
Okay, I think we're running out of time.
Can I ask one more? I'll take the liberty of asking the last question. For both of you, what are you really excited about? What are you looking at next?
Not being here. No. Okay, I mean, for me, if I can answer quickly, I think I'm excited about the fact that things are really coming together these days, right, in the field. And we had a discussion about consolidation. I think the field is very dynamic. It should be diverse. I mean, I'm not for a big consolidation of everything, but there's a lot of energy in putting different things together, and we see that, with gene editing and gene transfer. We see that, there are many instances of that. So there's a lot of things being invented, new thoughts. It's not a field that just is waiting for a solution, or so that keeps me excited.
So, two very short answers. As a physician, to be able to see that parents that are born with a kid that has a lethal disease have a solution to that, and those that have that opportunity, there's so much hope. If we could translate that to reality, I think it would change. We're all humans, and I think that's probably why we all do this. And I think we're there. As a scientist, I want to see if there's, with genome editing, when we go in vivo, how close are we? Are we really that close? And the 30 years it took us to get to where we are, it's going to be 3? Or are we really just another 20 years? And I'm not so sure, but I'm really anxious to get there to see where we are.
Okay. Well, thank you very much.
Yeah.
Thank you, Jim.
Okay.